Right, good morning, good afternoon, good evening, depending on where you are, and welcome to today's session of Soybean 360 Symposium. So we're glad that you were able to join us today, and we have a lineup of very fantastic speakers on a quite interesting area of soybean protein research. So the title of today's symposium is Innovations in Plant Protein Technology. So today we have a number of lectures. So I will start first of all by introducing the session, and then that will be followed by a quick introduction of the speakers. And so I believe you received the schedule already. So after each lecture, we'll have time for questions for each speaker. And that will be followed by a panel discussion where all the speakers would come. So in the process of the presentations, please, we encourage you to submit your questions so that your questions will be read to the speakers at the end of each presentation. So the emerging trends in research in the area of plant protein technology is quite advancing. So from today's lecture, you get to see a peek of some of the cutting edge work done at the interface between academic industry research, as well as from a government research perspective in plant protein technology. So without further ado, I would start with the introduction of the first speaker. So for the first speaker, we have, the title is Protein and Starch Interactions to Create Structure in Plant-Based Foods. And the speakers for today's, for the first lecture are Stacie Dobson and Dr. Alejandro Marangoni. So Stacie Dobson will be speaking, and I would quickly introduce her. So Stacie Dobson is an M.Sc student at the University of Guelph, Canada. And she is supervised by Dr. Alejandro Marangoni of the Food, Health, and Aging Laboratory at the University of Guelph. She's in the final stage of her master's program, where she has been developing a novel process for the creation of a fibrous meat analog. The research has allowed her to investigate the functionality, structure, and interactions between various plant proteins and starches. Stacie has also has certificate as a professional culinary chef, which has helped her further understand the food industry and create a product with desired taste and texture. So welcome at Stacie, and the floor is now yours. Thank you.
Okay, thank you for that introduction. Today's talk is going to be on protein-starch interactions to create structure in plant-based food. To start off, I'm going to talk about a bit of the reasoning behind the research we're doing and what has pushed us to pursue this plant-based industry. Sorry, it's not letting me move forward. Okay, there we go. So animal protein makes up a pretty large sector of the food industry. However, it's considered to be one of the most environmentally harmful and kind of unsustainable food sources. And with our growing population, it means we're going to have an increased demand for this kind of food. And with an increased demand, it means that all of the environmental factors that affect us right now are just going to increase. So the greenhouse gases associated with this industry, which are direct contributors to global warming and environmental damage, are just going to get worse and worse. So we're needing to find a solution. And when we look even at some statistics, we see that the beef, lamb, and goat in particular have some of the highest greenhouse gas emissions per gram of protein in this red region. And also the retail costs for these are incredibly high. And with the next following category, we have poultry and pork, where we see also the next highest greenhouse gas emissions per gram of protein, and also the relatively high costs. However, when we look into some plant-based alternatives for protein, such as soy or beans, chickpeas, lentils, they have quite low impact on the environment, and also their price is incredibly low, meaning that if we can make products out of these foods that would be more sustainable and ultimately cost less, then we could help start saving our planet and make more products that are sustainable. And when we look at specifically here replacing beef with plants in the US, we have a decrease of 90% across and greater across croplands, greenhouse gases, nitrogen fertilizers that would all create this decrease and make these products more sustainable. So there are products on the market that do this. Their meat analog industry is growing. We have the Beyond Burger, we have the Impossible Burger, we have all sorts of plant-based nuggets. Tofurkey has been in the industry for a while. And while these are all great alternatives, the problem we have is none of these are whole muscle meats. There's no steak, there's no chicken. And if we're going to start to want to grow the products and reach out to more consumers, we're going to have a variety of products that people want. So with that, we need to come up with a solution for that whole muscle fiber, that whole muscle meat, sorry. And also with these kind of patties and ground beefs, a lot of it is little kind of filamentous particles that then have to be stuck together with starches and fats and additives to get that texture and to get them to stick together. So all of those little pieces that are involved in making these grounds take a lot of energy. They take a lot of, it's not just a simple process. You can't just, grind up some soybeans or grind up some peas and add them in. They're going to have to be processed in a way to give us that fibrous texture. And specifically, we see TVP, or texturized vegetable protein, is a main ingredient in all of them. While it does provide a unique texture, that filamentous kind of ground beef texture, to make TVP, it's quite an extensive process. And specifically, it uses a lot of extrusion, which can have some problems when it's as it's highly processed. And so when we look at finding a whole muscle meat alternative, we're pretty limited. And so currently, it's either you have to find a plant that is fibrous, such as like jackfruit is pretty common as it can be pulled apart. It's kind of similar to, people compare it to more to pulled pork, or mushrooms are kind of growing in the industry. But with those plants, you have that limited nutritional value and that limited protein content, meaning that you're going to have to add in other products to build up that nutritional value. Or we get products where we see where we can get fibers through extrusion or shear cell processing. In these pictures here, we can see a shear cell steak, and or this small piece of extruded protein. However, in these shear cell processes and extrusion, it can be kind of problematic as there's incredibly high amounts of pressure and heat that go and kneading and mixing that are involved in this process. And while it can create a unique product, we have to think about the entire process and what it's doing to the actual protein itself. So when we think about extrusion, when I've looked into it, I mean, it creates a filamentous product. However, you're kind of limited on the size as you're left with that cold die setting at the end that is pretty much that main component which creates those layers. But that limited size means you're going to have to stick all those pieces again together to create that whole muscle meat or that large piece of steak that you're after. And the equipment to make this is quite expensive. There's large startup costs, meaning that you're going to have to throw a lot of money into this when when the whole process is kind of, I want to say trial and error, but it's extensive to determine the exact pressures, temperatures, to figure it all out. And then with the added temperatures and the added shear, we kind of makes us think like, what's happening with the Maillard reaction, as that increased kneading and shear can expose new reducing sugars and increase the Maillard products. And then all these products at the end have additional finishing steps, meaning that they're going to have to be cooked again and browned again, which, I mean, you're kind of cooking this protein until what nutritional value is really left is what makes me think. And that product is pretty tough. As I've had some people, I've tasted some myself, and it's more of a beef jerky than a tender steak. And to get that tender steak, it involves marinating and soaking for long periods of time to get that moisture back in the product. So that kind of leaves us with, what can we do to fill that gap? And our goal was to develop a novel meat analog that had a fibrous texture to mimic whole muscle meat, extrusion-free processing, plant-based, and sustainable. And we came up with a product that uses a prolamine, a protein isolate, and a rapid swelling starch to create this novel meat analog. So first, I'm going to talk about the filamentous portion of it. And we discovered that the prolamine, or the prolamin, was best suited for that. So prolamin proteins are hydrophobic proteins. They're found in corn, they're found in corn, which is zane, wheat, gluten, barley, horden, sorghum is caffeine. And so these hydrophobic proteins, a lot of them are considered waste products in the refining process, as they do have limited nutritional functionality because they are so hydrophobic, and also so they don't have a complete amino acid score. So they're going to need a protein to supplement that. However, they have very unique structural properties, as when they're plasticized under the correct conditions, we get these fibers that can just be stretched by hand. And when we look here with the addition of water or urea or salt, we don't see quite extensive fiber development. But when we look, when we look into the addition of an organic acid, we can see that these linear fibers that are able to be extended just manually without the need of any equipment, and they form filaments. However, we kind of realized, sure, these prolamins can form these filaments, but there's going to need to be a way to support them. And that's where we looked into a supporting network for these filaments. And in that, we determined that a rapid swelling starch and a protein isolate was our best bet. So the rapid swelling starch and the protein isolate is kind of like a combo matrix, in the sense that the rapid swelling starch, when hydrated in a pretty, let's say, a limited water condition, it likes to stick together. So all the starch molecules are kind of fighting for water, in a sense, meaning that they're all kind of stuck together and they make this dough-like structure. And so because it's this dough, very extendable structure, it's able to be manually extended and kneaded at the same time as we develop those fibers. And it actually is able to separate the fibers into a more fine fibril network. And with the addition of the protein, it actually has a structure-function relationship and changes how that starch kind of interacts. It disrupts the structure in a way and actually allows for different mechanical properties, and almost enhance the extendability and malleability of it with the addition of that added nutritional value. So together they form this unique supporting matrix that we feel is a unique structuring agent in foods that hasn't really been looked into extensively. And when we kind of look at some of the data, we can see that the confocal here, this is increasing protein amounts. This is 30%, 50%, 70%. The protein is actually acting as a particulate filler, in the sense that the protein is actively filling that continuous starch phase. And we can see that the protein is changing the mechanical properties with no protein present, we start to increase the amount of protein. The protein is acting as both a solid particle, increasing the hardness, but also kind of disrupting how the starch is, so it's not so stuck together. And then we decided that the hardness and the resilience are kind of two of the most important factors, as in order to develop those filaments with the prolamin, they have to be stretched, meaning that our supporting network is also going to have to be stretched and extendable. And we didn't want it to be extended and then kind of lose its shape. We wanted to keep its structure, meaning it's going to have to be incredibly resilient to endure that manual manipulation. And we see that the addition of protein does disrupt some of that resilience. However, finding the blend between the amount of protein added with the amount of starch, we can still keep high levels of resilience and be able to have that added nutritional value and also have that manipulative dough in itself. And so when we kind of look at how the that supporting network is able to interact with the addition of the fibers, we see some unique results. As I mentioned previously, the protein kind of disrupts the starch and allows it to be more extendable and get in between those fibers. And so having the proper ratio of that starch and protein, we can see is pretty essential, as ratios 2, 3, and 4. The black bars here is just the starch and the protein, with the gray bars being the addition of the fibrils. And so we see that there's not too much difference. It's not adding very much structure, but in our ratio 1, we're able to have a pretty high hardness with just the protein and the starch. And actually the addition of these fibrils, it enhances the network, it changes it, it makes it harder. At this point, the starch and protein are interacting in a way that is allowing these filaments to be separated and almost create a unique network in itself. And when we look into the other mechanical parameters, we see the chewiness and the resilience. The starch and protein by itself, the black bar, we see that it has generally high chewiness and high resilience with just the starch and protein. And the addition of the fibrils, it starts to decrease it, which is actually the feature that we were looking for, in the sense that when you kind of bite into a piece of meat, it's not like a piece of gum or not like a piece of bread. You want to be able to bite through it. And so we don't want high amounts of chewiness. We don't want it to bounce back like a resilient dough. There's some bounce in meat, but a lot of it is once these fibers are cooked, they're they keep their shape, and you can able to bite through them and have that more desirable mouthfeel. And so the addition of that fibril network, it changes those textural properties, which is a pretty unique feature in this system. And when we look at the amount of, and then so also we looked into how the fibrils play in this network. And we found that there had to be a balance. So the starch protein network, it can only hold so much. As soon as you overload it, we're left with a fibrous network that kind of clumps together. Sorry. That kind of clumps together as the analog. It becomes too packed with fibers, and there's not enough supporting matrix to separate them, and they tend to clump together and actually reduces the mechanical properties, and it loses elasticity, and it becomes a more brittle network. But on the other spectrum, when we see down here, when we add too little, there's not enough prolamine, we don't have there's not any fibrous network. It's more like a bouncy dough. So finding that blend of which we have the ideal amount of fibrils added was also a part of the study we were doing as well, to determine what was the almost maximum we could add and what was the minimum we can add. And actually the amount of fibers you add, it again changes those textural properties. So you can almost make a product that, let's say, is more like beef or more like chicken or more like fish for that. I mean, I wouldn't say fish, but or pork. So each of those have different fibrous qualities that you can change based on the amount of prolamine you add. And when we look into some of our confocal here of the fibrous network in that supporting matrix, we can see those linear fibers running through it, which in the background, we can see those, the protein isolate is also present and embedded, and with the black background being the starch, and that it's a unique structure-function relationship network that's holding all of this together and creating this neat analog that can be made with your hands, doesn't need expensive equipment, and it's more sustainable in the sense that we're using a product that people find as a waste. And we get a final product that browns just like a meat or any product that you're looking to cook. And when we look into a slice of it, we can see those fibers running through it, and that the supporting starch protein isolate network is actually kind of, it's still intact. It's suspending those fibers, and it's allowing the product to keep the fibers, but also break in a way that has that filamentous texture. And so we think that this product is pretty unique in the sense that we haven't seen anything else like it, and it doesn't involve these expensive machinery. And we're able to just make a product that is quite unique. And so again, the research that we have done is definitely at a lab scale, and eventually we may look into scaling it up or trying to figure out other proteins that could be utilized in this unique manner. And that's kind of it for the presentation. Thank you guys for listening. Dr. Marangoni, do you want to add anything in? Oh.
No, thank you very much. That was great.
Okay, thank you, Stacie, for that excellent presentation, and I quite enjoyed it. This is time for questions from the audience. But while we wait for the questions, I have a quick question about the implication of the protein-starch interactions that you mentioned. So now the purpose of the interaction is to create a functional network that would mimic the meat structure, pseudo meat structure. But at the same time, this is also intended for nutritional purpose. So what do you expect will be the outcome of these interactions in terms of their effect on digestibility of both the starch and the protein?
Okay, good, perfect. So I don't think there's going to be quite any implications on the digestibility, as the products that we're using, the prolamine itself is regulated for consumption. And also we're using a protein isolate that is readily available in the sense that people can make their protein shakes out of it, or people use them in other foods. And the starches, in the same way, it's all regulated for consumption. So we're not too worried about how the digestibility is going to be affected, but we're kind of more focusing right now on those textural properties that people desire in food, as I think having those sensory and textural properties is kind of one of the, and making this product that a consumer wants is important, because people want that desired mouthfeel and those desire, and people are more after those characteristics when they first bite into it. So again, developing this method also isn't limiting us to introducing other proteins potentially in the future. So there's ways to change the digestibility or functionality in that way as well.
If I may add, one of the reasons for us going into this, we were a little bit concerned, Chibuike, like you were saying, about the nutritional qualities of extruded protein. Under high moisture extrusion, usually you have to heat up the protein to 150 degrees under high pressure and shear. And I always wonder whether there were undesirable cross-linking reactions or that would limit digestibility or destroy lysine or things like that. All of these processes are taking place at 60 degrees Celsius. So we're even below the denaturation temperature of the protein. So this is just a physical mixture, and the nutritional quality of the original material gets preserved into the final product. But you do have to remember that these prolamines usually don't have a very high nutritional value. They're strictly there for the textural properties But the majority, the bulk of the nutrition comes from the protein and the starch. But I think what we found in our experience is you have to be really careful choosing these ingredients. The type of starch and the protein plays a huge role, and the way it gets processed. Many different isolates didn't work, only a certain isolate worked. So I think there's a huge room here for people to really characterize the functional properties of the protein isolates that are available commercially, because there's a huge variability in both protein quality and the starch. Even two starches that are labeled the same are different. We really need to do a better job at characterizing those materials in terms of nutrition and functionality.
Okay, thank you very much, Stacie and Alejandro. We have a question from the audience. One question is, what would you predict the addition of fat will do to your system?
So that's a interesting question. So with the addition of fat, I think that it could create a unique product in the sense that the prolamine may plasticize differently. However, introducing it in a way that the fat remains solid, I don't think is going to remain solid when it's kind of set and cold. I don't think will break the structure in any way, as it's there's strong interactions keeping those starches and proteins together, as it is a limitedly hydrated network. So adding in another type of fat, or in the sense you're adding in another particle, I think we would just have to adjust the amount of starch to protein ratio to kind of account for any type of structural loss that may happen with the addition of fat.
You inherently increase the lubrication of all the structures there. We tried, right, Stacie, a couple of times to include something. Everything starts slipping. So you do change the extensional rheological properties of the material with it. So you have to reformulate again, because remember, you need a balance of the elasticity, the hardness, the resilience of the supporting network, so that you get the extension on that prolamine. You start changing that, you're going to change the processes. But yeah, fat, I think it plays a huge role, and we're going to have to explore that carefully. The system changes. So yeah, you lubricate everything. So now you're changing the extensional rheology, which is what creates those fibers. So that's a great question, and we'll be looking into that this coming winter.
Okay, thank you very much, Stacie and Alex. So a last question that we have from the audience is about the shelf life. So the question is, how can you compare the shelf life of the product compared to traditionally extruded products? Is there any difference?
So we don't really have any concerns about the shelf life, in the sense that the product is actually made in a low pH system, as the addition of the organic acid is needed to have that plasticization to make those fibers. So we're working at a pretty low pH already. So having that limited pH kind of limits the type of growth or microbial issues that we would have with that product. Shelf life wise, and the ways that I've made it, I vacuum seal the product just like most people would do in their package. And once I've also previously frozen it, and you can take it out of and let it come to room temperature, and you still regain that the elasticity of it, as when it again is heated like any other steak or whole muscle meat. The heat comes through it again and actually regains some of that elasticity within the product. So shelf life wise, we don't have too many worries due to that low pH and the methods that we're using to make the product.
Okay, thank you very much, Stacie, and thank you, Alejandro. This is a very interesting topic, and I'm quite excited to see this opportunity to discuss it. So we'll reserve any further questions for the panel discussion, and so I would like to thank you once again, Stacie, for that presentation, and Dr. Marangoni for also sharing your insight during the question and answer. Thank you very much.
Thank you.
Okay, we have a second speaker, Dr. Michelle Braun. So a quick introduction of Dr. Braun. Dr. Michelle Braun is the Global Protein Scientific Affairs Lead for DuPont Nutrition and Biosciences, United States. She's a nutrition scientist with experience in clinical research and nutrition translation. Dr. Braun is currently the Global Protein Affairs Lead that I had mentioned, and over the course of her career, she has led research investigating a variety of health outcomes with a particular focus on child nutrition. Dr. Braun is also involved in research conducting several clinical trials related to protein and mineral metabolism in children. She's active in several organizations related to the field and has served in volunteer and leadership roles in the American Society of Nutrition, the Institute for Food Technologies, the International Life Sciences Institute, and International Food Information Council. And she's also the current president of the Soy Nutrition Institute. Dr. Braun has earned her doctorate and master's degree in foods and nutrition from Purdue University, USA, with a bachelor's degree in nutrition from Indiana University. So today, Dr. Braun will be talking about delivering high quality protein in a variety of food formats. You're welcome, Dr. Braun. The floor is yours.
Hello to everyone. It's my pleasure to join you today. I appreciate that excellent introduction. And just briefly, I just want to acknowledge, yes, I am with DuPont Nutrition and Biosciences, and that company is a provider of a variety of food ingredients, including the Supro line of plant proteins, for full disclosure today. As we begin, we would need to acknowledge that the plant-based space is getting very crowded with lots of options. However, there are quantity and quality considerations that we must consider in terms of human health. Quantity is possibly the simpler concept, even with the debate of how much protein do we need, because protein is an essential nutrient, meaning we need it every day to support healthy functions of at the cellular level and tissue level for the entire body. The quantity of protein available is different in the sources, in different sources, as shown here. Just as the quantity of protein varies in finished foods that we consume, as is shown in the next slide. This is a list of common high protein plant-based foods. As you can see, legumes, nuts, and seeds tend to have highest amount of proteins per serving. Vegetables and grains can provide protein as well, although not to the same extent as legumes, nuts, and seeds. There are also food products made from plant ingredients, including soybeans, that will deliver plant proteins such as tofu and soy milk, which are more traditional food forms of that are based in ingredient versions of plant sources. But we know that this is an exciting area, and we heard from the last speaker of the new food formulations, the new food options that are emerging in the marketplace. So it's an exciting area to keep tabs on, to consider how plant proteins can be a part of our diets and end up on our plates each day. So protein is essential, and it needs to be included in the diet every day. And clearly there's a variety of options that can contribute to daily goals, which are generally set at just a shorthand way to think about it is just under 1 gram of protein to fuel the needs based on each kilogram of body weight, or technically 0.8 grams per kilogram body weight. Now, when we think about when we move past protein quantity into protein quality, there are differences in the ability of protein sources to meet those needs, which are based on the amino acids present in the protein and how well digested the protein is digested and utilized by the body. Proteins are made up of different combinations of amino acids. There are 9 amino acids that must be obtained in the diet. The protein digestibility corrected amino acid score, or PDCAS, is the method of protein quality measurement based on the principle that the nutritive value of protein depends on its ability to provide amino acids in adequate amounts to meet the requirements of children and adults. And it's a method for protein. It is the method for protein quality measurement referenced in many of the current nutrition labeling guidelines. And it's used to describe the quality of the protein or its ability to meet the needs of the young and the old. But let's take a look at that. This is the essential amino acid needs as described by this method. And we see here the needs of the very young and young children. PDCAS method uses the needs not of rapidly growing infants, which is a state of metabolism distinct for that time in life, but rather growing children. And it's a conservative measure. If a protein source can meet the needs of children, then it can also meet the needs of adults. And this pattern of essential amino acid is needed to fuel healthy growth and development of this age group. The next slide shows how one plant source or soy protein stacks up to those requirements. As you can see, soy meets the needs of children and thus adults by supplying the essential amino acids in the right proportions to meet needs. Digestibility is taken into consideration in the values that are shown here. And isolated soy protein has been shown to be, on average, about 97 to 98% digestible. So it's well digested. It's expected that the more isolated forms of any ingredient or plant source can increase in digestibility, through processing due to the reduction of the inherent digestion inhibitors. So many plant sources also have, along with the protein, fiber and some factors that are considered anti-nutritional factors, such as protease inhibitors, tannins, phytic acid, etc. This makes, but the processing to remove those components in the isolated forms, and testing reveals through the PDCAS method that the quality of soy protein, as assessed by PDCAS, is comparable to milk and egg protein. As a plant source, isolated soy protein is also low in fat, saturated fat, cholesterol-free, and lactose-free. And as shown here, it can be consumed throughout the life cycle. Now, it's recognized that plant proteins do not support health in isolation. It's more than just the quality of the protein. It includes, plant sources include healthy fats, fibers, antioxidants. These are, we get so many nutrients from plant sources. Over the past decade, there's been a shift in nutrition guidance from focusing on single nutrients, such as reducing saturated fat and sodium or increasing fiber and potassium, to now concentrating on dietary patterns. This shift likely occurred for several reasons. Nutrients are not consumed in isolation, and therefore interactions between foods are missed. And the importance of dietary patterns that combine various foods and nutrients can't be overlooked, related to their significant effect on health and in a way that's that can be well digested as we translate nutrition and diet guidance to the public. Therefore, global health authorities are moving away from nutrient-based recommendations in favor of dietary pattern-based recommendations, and diets rich in plant foods, fruits, vegetables, legumes, whole grains, nuts, seeds, soy products, and vegetable oils are increasingly being recommended by many authorities, as shown here, due to the strong evidence supporting the beneficial health effects and associations of higher consumption with lower risk of diseases such as cardiometabolic disease. So dietary recommendations based on high quality research that substantiate many aspects of health can support the inclusion of plant-based foods in the diet. And soy plays a significant role in many of these well-studied dietary patterns. And here's just a short list. Dietary patterns are frequently recommended by international dietary and clinical practice guidelines, and especially when we think about heart disease risk. So a few of the diets that include soy and are well studied for their health effects are listed here, including the Mediterranean diet, which is high in fruits and vegetables, whole grains, legumes, nuts, and olive oil. This has been well studied in terms of its effects on persons who are at high risk for cardiovascular disease and its efficacy. The DASH diet, or the dietary approach to stop hypertension diet, also recommends 4 to 5 servings of plant sources per week. The low GI diet, or glycemic index diet, may be popular where you are. And of course, the vegetarian diet, that's fairly straightforward, that plant sources would be endorsed within the vegetarian diet. And it's important to recognize that soy is an essential high quality protein source for vegetarians to help meet their needs in a single source and help to bridge any gaps that may come through adhering to that diet. And the list goes on. So a common theme for these dietary patterns is that they are primarily plant-based, and soy can play a significant role within them. So we're talking about optimizing health, but we do need to recognize that there's significant evidence showing that dietary guidelines are not being met in many countries and consumer populations, while overconsumption of foods high in saturated fats and added sugars is contributing to the increasing prevalence of overweight, obesity, diabetes, and heart disease. There are nutrition challenges that are faced in many parts of the globe that can contribute to deficiencies of key nutrients such as vitamin A, iron, zinc, and protein. And this can be compounded by digestion challenges such as lactose intolerance, which make foods that deliver many of these nutrients from dairy sources a less optimal solution. So let's just underscore the importance of these nutrients that were highlighted on the previous map. For example, lactose intolerance is thought to impact about 65% of the world's population, causing gastrointestinal distress and bloating. Vitamin A affects 25% of preschool children and pregnant women in developing countries and can lead to difficulty in seeing in low light or to the extreme blindness. For iron deficiency, 30% of the world's population may be impacted, with 50% of pregnant women and 40% of preschool children in developing countries. Signs and symptoms of iron deficiency include anemia, weakness, fatigue, and shortness of breath. And 17% of the world's population may be impacted by zinc deficiency, which is associated with compromised immunity and stunted growth, to name a few. Possibly less well recognized is protein energy malnutrition. More than a third of the world's children can be impacted by this condition. So there are plant-based products that can help to address these concerns by contributing these important nutrients. Oops, I just clicked away. Here we go. And here's an example. Now, I know this is a busy slide, but it includes the nutrition data for a product that can function as a cow's milk alternative. The nutrition profile shares many key nutrients found in whole milk. The products are compared on a per 100 gram basis. So we have whole milk powder, whole cow's milk powder listed in the first column, followed by 2 soy-based, fortified soy-based ingredient offerings. And one way to think about how to compare these on a per serving basis is to roughly divide by 3, where a glass of milk, cow's milk, delivers about 8 grams of high quality protein. And if we divide the other 2 products similarly, we'll see that the fortified soy-based alternative also delivers at least 8 grams of protein per serving, along with a meaningful amount of vitamin A, zinc, and calcium. So this is a viable option for addressing some of those nutrients of need and concern. And this ingredient can be used in a variety of foods or eating experiences, from a traditional beverage that can be reconstituted with clean potable water, but also can serve as a nutritious base for desserts, yogurts, frozen desserts, and soups. So it's nice to have those eating options and the variety in terms of the different foods that can manifest on the plate for families to consume. The powdered products aren't the only way that soy ingredients can be available. Soy ingredients can come in a variety of forms and formats for inclusion in recipes or formulas of new or familiar foods. Powders do deliver application-specific functionality for beverages. So whether it's a dry, product that's to be reconstituted or ready to drink or spray dried, the high quality protein can come through in the powder products. These are also often used in meat or poultry or meat-free applications as well. So this is the most versatile form that can be used in general protein fortification. But soy protein has been in the marketplace long enough that there are many other forms and formats that are available. Extruded products such as nuggets that can add crispy or crunchiness to an eating experience, such as in a bar or cereal or snack, can deliver high quality protein, where a lot of times we think of a lower quality protein, such as a rice crisp being in that space, can be swapped out for a protein crisp. And other extruded products can deliver different eating experiences. Textured soy protein concentrate is available in flakes and granules and crumbles, which works well in meat and poultry or meat-free versions of those applications. And structured vegetable protein can mimic the whole muscle texture, whole muscle-like texture in shreds, chunks, and strips formats. And of course, you know, as we heard in the previous talk, this is a very exciting area where soy has a head start in terms of the innovation that has been applied and what we know of how this ingredient can manifest in a variety of foods and formats. So let's return to the topic of protein quality and discuss for a moment the applicability of this method. Now we're going to switch into the health aspects of the talk, and I will try not to overstay my welcome. In terms of the tool PDCAST, we've talked about how soy is a high quality source meeting the needs of children as well as adults. And as you can see in this chart, PDCAST method reveals that soy is a high quality source of protein similar to milk and egg. When a protein source is lower on the chart, it's likely due to the presence of a lower amount of one or more essential amino acids, meaning those amino acids that we must obtain in the diet. The protein source may also be lower in digestibility, and we've talked a lot about that. So we've also recognized that protein is an important nutrient essential in the diet to fuel healthy growth and development. But children who face food scarcity challenges and develop severe acute malnutrition need safe, palatable foods that have energy, protein, fats, minerals, and vitamins tailored to meet their needs to recover. So now we're not talking about optimizing daily health, but how can we assess whether a protein source can help to recover from severe acute malnutrition? Now, globally, 20 million children suffer from severe acute malnutrition, but with proper treatment can grow up to lead normal or productive lives. Several years ago, DuPont Nutrition and Biosciences partnered with the United Soybean Board to develop and test a high-quality soy-based recovery product. Researchers in this field were seeking treatments and investigated the efficacy of the product that was soy-based in comparison to the traditional milk peanut-based product to support recovery, provide high-quality nutrition, and investigate whether this may be a more cost-effective treatment for affected populations. And this was compared to the standard of care. In treating severe acute malnutrition, the World Health Organization recommends energy-dense liquid peanut paste, sorry, that's a mouthful, that are enriched with vitamins and minerals and include those high-cost dairy ingredients. These pastes deliver much-needed high-quality protein, but often pose tolerance and digestion challenges in affected patients. So I have an image of what our product essentially looked like in cartoon at the bottom of the slide. But what's important is whether a product, it's not just to be able to make it, but does it support recovery? The researchers found that the soy-based ready-to-use therapeutic food was equally acceptable as the milk-based RUTF, or ready-to-use therapeutic food. Plus, the children who received it showed similar patterns of weight gain, rate of weight gain, and other positive changes in anthropometric measures and body composition as those receiving the milk-based ready-to-use therapeutic food. So this study demonstrated that the RUTF made with high-quality soy protein can be an effective treatment for children with severe acute malnutrition. So this is really the manifestation of protein quality. A protein may be considered high quality in terms of the PDCAS method, but can it equally support other recognized high quality sources of support recovery as other high quality sources of protein? The answer to that question is yes from this study. And we also found an alternative that may be more well tolerated for the treatment of children with severe acute malnutrition. So this study was significant because it provides evidence that rescue foods for children with SAM can be more affordable, yet equally effective. Now, let's talk a little bit more beyond the needs of children who are suffering from severe acute malnutrition to the evidence that has been established on how soy can support health and many aspects of health across the lifespan. Soy is probably the most well-studied plant source of protein, where research substantiates its effects supporting healthy growth and development, which we've talked about. But also soy has been shown to support several aspects of weight management, lean tissue, and what may be best well known due to the hundreds of studies investigating this area are the heart health effects that have led to numerous claims in countries around the globe. So let's begin by focusing in on those heart health benefits. Soy has been demonstrated to support several aspects of heart health, from reducing risk factors associated with cardiovascular disease or direct reduction of development of cardiovascular disease, to other aspects of health maybe that we think of earlier in life as supporting optimal health of blood vessels. Really, this is a robust area of research. And we're going to talk about each and every study. No, I'm just teasing. I am a fan of sharing meta-analyses, and there's been so much research in soy and heart health that there's actually been multiple meta-analyses. So we're looking at the totality of high-quality studies that are available at a particular time when authors conducted their meta-analyses of looking at the totality of the evidence. What's similar across these studies, and you can see I have 7 studies selected at the top of the slide. What's similar is that each of the authors considered the risk factors for cardiovascular disease as their health outcomes: total cholesterol, pictured in blue, LDL, or the bad cholesterol, pictured in red, triglycerides in purple, and HDL in green. And these are also some of the outcomes that are considered in the health claim dossiers that are in place in over 27 countries, in over 12 countries around the globe. So what's notable from the outcomes of these meta-analyses is the agreement across the different studies that no matter when the authors selected their studies or the quality criteria that were considered, each of these studies demonstrates that soy protein can lower total cholesterol and LDL cholesterol and triglycerides without lowering HDL cholesterol, which is actually quite notable. So many studies have been conducted on soy and heart health that we have this strong foundation when we talk about the heart health benefits of soy. Another health outcome area that's of interest and to consumers, because they think of it more about an immediate need, and that's a product's ability to help to support their weight management goals. What's interesting for soy is that the research that's been conducted shows that soy is effective both in the short-term aspects of weight management, which is notably assessed in terms of satiety or the feeling of fullness. So several studies have demonstrated, like other high-quality sources of protein, soy can support satiety and also can help to reduce, by consuming soy at one eating occasion, it may help to impact caloric intake at the next meal over the course of the day. Now, if we were to repeat that for multiple days, then we get to a longer term intervention such as weight loss, which soy has been well studied in terms of its effects in supporting weight loss in comparison to other high quality sources of protein. And really those studies are considering what's happening for the duration of the intervention. But what happens when the weight loss study or the weight loss program concludes? Well, that's when we hear a lot about the yo-yo, or people regaining a lot of that weight. So there's been research that assesses soy's ability to support weight maintenance, or the ability to maintain that weight loss. And studies show that while some weight regain does occur when people are free living and no longer a part of a program where they know that, that they're working towards that goal, what is observed is that the weight regain is not in fat tissue, but in lean tissue. So there's definitely some impact there on the lean tissue or muscle tissue in terms of protein's ability to drive accretion of lean tissue. And then, of course, by including soy in a weight loss intervention or weight loss program, it can help to support lasting or long-term effects in terms of cardiometabolic health. And that relates back to the heart health benefits, which we've well discussed. So let's drill down on that lean tissue or the muscle health effects. This will probably be the busiest slide that I show you today. But again, it's a meta-analysis. So we hear so much about the effect of proteins on muscle. And a lot of that attention is driven by the acute studies or the very short-term studies that look at how muscle is metabolizing and utilizing protein sources in the short term after exercise. This meta-analysis, I really like this one because it only takes into consideration those of longer terms, the chronic studies that compare soy to other high-quality sources of protein, which are animal sources. And we are looking at health outcomes related to lean body mass gains and strength. And the blue diamonds, since they are right aligned with each other, demonstrate that there are no differences between soy and whey protein or other animal protein sources that were taken into consideration in this study for support of these health outcomes related to muscle. So the conclusions that soy stimulates muscle protein synthesis or supports muscle health to a lesser extent than whey protein is really driven by those acute studies, and it's a false conclusion as demonstrated here in this research. Oh dear. And finally, it's an exciting area of research still to be in the business of uncovering how soy can affect health as we think about our current challenges that we face in terms of obesity and how the gut microbiome may interplay with some of the emerging areas of investigation, including glucose and insulin metabolism and other aspects of metabolic health. So lots to keep an eye on in terms of research related to soy and health. So in conclusion, there's evidence to support that the inclusion of plant sources in the diet can support health and optimize health, but also address risk factors associated with conditions that affect many people, whether it's severe acute malnutrition for children or heart health for those who are at the other end of the lifespan. Due to differences in composition, different dietary plant protein sources may have differing effects on health. We just don't have as much research on other plant sources as we do on soy. And diets that include plant sources can be adapted to local traditions as ingredients that are soy-based can take a variety of forms and formats, but it's important to still be mindful of including sources that are demonstrated to support health. And with that, I will conclude, and thank you for your attention.
Okay, thank you very much, Dr. Braun, for that excellent presentation. So you have quite a number of questions from the audience, but I also have one question. So we'll start with the audience questions. The first question is, are you aware of any in vitro or non-animal testing alternatives for measuring PDCAS?
That's a great question. This is there's very much a changing landscape in terms of the assessment of protein quality. And there is an outcry from many consumer groups to avoid animal testing. It's interesting because protein is, in many regulatory jurisdictions, protein is the only nutrient that we must report that's based on animal testing. And so there are viable in vitro methods that are that have been in place for a number of years, but not yet adopted by regulatory authorities. Therefore, they are often utilized by manufacturers, but they are not necessarily 100% in line with regulations. But we are caught between a dynamic where we're moving closer to animal methods if we were to adopt, say, the DIAS method using the swine model, and consumers wish to avoid animal testing. So this is really an area that requires a lot of attention in the years ahead, and recognition that for now we are tied to the PDCAS method, which uses a rat digestibility method. Great question.
All right, thank you. So the next question is not particularly on proteins. It says, is there a research scale that your company will be interested in evaluating soybean seeds with enhanced seed composition for carbohydrates and oil? So is there a research scale that your company will be interested in?
I'd be happy to have a follow-up. I'd be happy to follow up offline with that individual, and we can discuss that. And if it's not us, it may be another partner in the supply chain. But yes, it's an exciting area for sure.
Okay. And then the last question is coming from me. So you presented different matrices through which soybean or other plant protein ingredients could be incorporated into food. So do you see any potential variation in terms of the quality of the protein delivered when using, say, a protein isolate or concentrate or flour in different matrices?
Well, we do know that the quality, as assessed by PDCAS, shifts as we talk about the more processed that or less processed. So soy flour has a lower protein quality than concentrate, than isolate. Concentrate and isolate, depending, the definitions of concentrate and isolate, you can, there's a band, right? So concentrates have been demonstrated to have similar protein quality to some isolates. But I think the previous speaker also acknowledged that there are differences depending on which concentrate. I think what you're also getting at is if we were to process those further into extruded products, how that might impact protein quality. Our testing thus far demonstrates that the effect in extrusion is minimal, and when we take an isolate into an extruded product, we still see high quality in terms of how PDCAS. But there's more research that's needed there. There's not as much data as we have for the flours, the concentrates, and the isolates in their native forms. We also have a paucity of data in terms of other food forms. So we know less about some of the other food forms that may be derived from whole bean and really where they fit in that lineup. I think a lot of times we've just been using the ingredient values, but that testing hasn't been conducted in the same manner. And as we just talked about in the previous question, we're in a shifting landscape in terms of methods for protein quality. Those data may be forthcoming in the future.
Okay, thank you very much, Dr. Braun, for your excellent presentation and answers to the questions. Okay, welcome to the third presentation this morning. So we have a speaker, Dr. Keshun Liu. So I'll briefly introduce Dr. Liu. Keshun Liu is a research chemist with United States Department of Agriculture, Agricultural Research Service. Born in rural China, he received his master's and PhD degrees in food science from Michigan State University in the US, and also did a postdoctoral work at Coca-Cola Company and the University of Georgia. And after that, prior to joining USDA in 2005, he was employed at Monsanto and the University of Missouri. Dr. Liu's expertise is in the area of chemistry, processing, and value-added utilization of soybeans. And this work in his lab has spanned over the last 35 years of his career at different academic institutions, private industry, and government agencies. Dr. Liu has co-authored over 137 publications, organized several conferences and symposia and scientific meetings, and has also given over 110 technical presentations at many conferences nationally and internationally. So he has, Dr. Liu has extensive experience as a journal editor and a book editor. And one of his books is Soybean as Functional Foods and Ingredients, published by AOCS Press in 2005. And his scientific achievements and contributions are also shown in his activities in different scientific organizations, including AOCS, and that has led to various recognitions, including AOCS Merit Award in 2010, and AOCS Fellow in 2011, and IFT Fellow in 2014, as well as the first AOCS Protein and Co-Product Division Lifetime Achievement Award in 2020. Dr. Liu today will be speaking to us about developing low-cost soy protein concentrates for expanding soy protein utilization. Welcome, Dr. Liu, and the floor is yours.
Yes, thank you so much, Chikubi, for your introductions. Again, my name is Keshun Liu. I'm with the U.S. Department of Agriculture Research Service. So today I'm going to talk about a project that been done a few years ago. So it's about developing low-cost soy protein concentrates. Oh, just 1 minute. Sorry. Okay.
So Keshun, you can try hitting enter or one of your arrow buttons.
Sorry, where the screen is?
You can also look down and you see on the right-hand side where there's some arrows on your PowerPoint file.
Uh-huh.
And you can click the one to the right.
Maybe the Zoom is gone. Let me see. Oh. Yeah, that's terrible. I don't know where this screen out. Where's the screen?
It's okay, Keshun. I'm going to get your slides up and I'm just going to advance them for you. Okay, so just give me 1 second.
I don't supposed to see this content, but right now I cannot see the content. Maybe I start over again. Okay, sorry. I just leave the Thank you, Chikubi, for your good introduction. My name is Keshun Liu. I'm with the U.S. Department of Agricultural Research Service. So today I'm going to talk about a project we was done a few years ago on developing low-cost soy protein concentrates. This is the presentation outline. I will give a very brief introduction on soybeans in terms of water production and the processing and utilization, soy protein products. Within soybean protein product, I will focus a little bit more on the soy protein concentrates in terms of opportunity and the challenges. Then I will talk on the project on developing low-cost soy protein concentrates in terms of strategy, method, product feature, and applications. And this slide shows the last 50 years of the world soybean productions from 1970 until this year. So you can see the soybean production increased steadily, particularly in the last 20 years. Currently, the world total soybean production annual production is estimated at 380 million metric tons. Oh, just maybe too fast. The Brazil is the leading soybean production. A couple of years ago, the U.S. is leading soybean production. Now Brazil surpassed the U.S. as the #1 soybean producer. And this followed by U.S., Argentina, China, India, etc. Now come to the questions. Why do we need to process soybeans? Well, soybeans are not like a rice, so we can cook this. Like we can eat cooked rice every day, no problem. But the soybeans, after cooked, is edible, but you cannot eat it every day. So we have to process soybean into various type of food, soy food or soy ingredients. Also, it's necessary to press soybean to reduce the eliminate beany flavors and reduce and eliminate certain anti-nutrients. Also increase the value of soy industry, so by processing and provide benefits to consumer. So consumer can have nutritious, healthy, and various tasty soy food. Soybean also contain many bioactive compounds. Some of them are being considered as have health benefits, but most others have been traditionally considered anti-nutritionals. So those bioactives have been catalyzed, generally catalyzed as a heat liable and a heat stable groups. So in terms of the heat liables, the two examples is the trypsin inhibitors, then the lectins, also called hemagglutinin. The heat stable is the one we will focus today. Is the two major one is the oligosaccharides, as shown on the top right, is the molecular structure. Major one, and sucrose, of course, is one of them. But the two actually major concern is the raffinose and the stachyls. Soybean contain about 3.5% of raffinose on the dry mass basis, about 1% a little bit high on stachyls. So they are together about 4 or 5%. So relatively high in the soybeans. So they are trisaccharides, as shown here, or tetrasaccharides, consist of glucose, fructose, and galactose. The other one is a fatty acid. It's also heat stable. Fatty acid is an inositol ester of phosphate. It is a major phosphorus storage compound in plant material, particularly in soybeans. I think it contains about 1 to 2% in the soybeans. So fatty can reduce mineral absorptions in animals, of course, including humans. In terms of soybean processing, we have traditional processing and modern processing. The traditional soy processing, which is created about 2,000 years ago in the Far East, they can be as a fermented soy foods, including like a soy sauce, miso, lettuce, also non-fermented soy milk and tofu, soy sprout. And some of these products have been in the Western market for many years. Here's a picture. You can see different tofu. Nowadays, I think certainly people are very familiar with some of these soy foods. Modern soybean processing is a major feature is the solvent extraction. So not going to get a detail about this process, and we have a speaker previously talk about this area. And the major products is the soy oil, which is mostly for edible applications. And then the DuPont soy meal. In the DuPont soy meal, we have two types of products. One is the water flakes, which is desulfatized at a lower temperature. I think we have previous speaker, Mr. Richard Wooser, talk about water flake production. Then the other is a major one is toasted soy meal, which is during this desulfatizing soy meal was heated, toasted. The most currently the mechanical processing, like a screw and hydraulic process, has been a very traditional method, but not really very much used for soybeans because this process is more for the high oil content oil seeds, like a rapeseed, some other oil seed. But for soybeans, a much more popular one is a extruding expression process. I think it was originally invented, created in the University of Illinois in the 60s. This process is also known as the extrusion-aided screw processing, pressing. So we have a few days ago, Dr. Wilma Wageningen gave a talk on this process. So both this alternative process produce oil and partially defatted soy cake. Now this one just shows the outline of the alternative processing through the called EE process, extruding and expanding method. So we start with oil seeds, then dehulling is optional. Then then dry extruding to disrupt this cell structure. Then also heat soybeans at different levels. So this 4-5 extrudates look like a very oily material. So my next step is expelling, so the press and oil out. This and then have crude oil. Then also the called partially defined soy cake contain about 5 to 10% of oil content, compared with originally about 20% oil content in the soybean material. Now, soybeans have been processed. The modern processing, the soy oil has been edible for edible application mostly, but the soy protein part actually currently major part is used for animal feed. But for human consumption, we process soybean protein cake into different type of ingredients. The 4 major types is soy flour, soy protein concentrate, soy protein isolate, and packaged soy protein products. One reason also is the traditional soy foods are not well accepted. Quite a few are still not well accepted. So those are mostly made from different soy, like white flakes. Some are from whole soy or partially different soy. I mentioned about soy cakes. So they are incorporated into various Western foods. So also, as we talked this with this morning session, have 2 to 3 speakers talk about meat analogues. So soy protein ingredients is a major ingredient for we call new generation soy foods, which including the meatless analog, meat analog, or meatless burger, or meatless steak, whatever. So this process, this chart shows how these three major soy protein products are made. So from left is soy flour. It's the simple one, just through particle reduction. And to the far right is soy protein isolate, which is this alcohol extraction and acid precipitation. And the middle one is soy protein concentrate. This is typically through alcohol extraction, leaching process. So from left to the right, there's more complexity in the processing. Also, the production cost is increasing. This one shows the three major soy protein ingredients in terms of the compositions. Compared with different soy flour, the soy protein concentrates contain, because soluble carbohydrate is reduced. So other composition like insoluble carbohydrates are similar to soy flour. The protein content increased to about 65%. But further, for the soy isolate, the insoluble carbohydrate and the soluble carbohydrate are both removed. So their protein content is much higher. We focus more for this talk about soy protein concentrate, because soy protein content have an opportunity. We can see the soy flour is not ideal for protein enrichment because they contain heat-stable anti-nutrients such as oligosaccharides and phytate. They can cause flatus. So the soy they also have a peanut flavor. So inclusion level typically in the food product is limited, mostly less than 10%. In comparison, soy protein content is better than soy flour because the protein content is high, significantly high, and contain less heat-stable anti-nutrients and less beany flavor. But soy protein content have some challenges. First of all, the price is between soy flour and isolate. So it is relatively expensive for some processor, in terms of the product development. So also the limited availability in certain areas, certain regions. So a couple of years ago, at the USDA, my colleague, Dr. Rick Barrows, now retired, and I took a project to develop a low-cost soy protein concentrates. So our objective is reduce the production cost while keeping the nutritional value as the same as the regular soy protein concentrate. This project was partially funded by Indiana Soybean Allies. Of course, USDA is the major funder for us for this project. This slide shows the principle of making soy protein concentrate. They start with the soy cake, soy flake or milk. Then we insolubilize protein while leach or extract soluble components, and keep the insoluble carbohydrate and the protein in the final product. So our strategy is identify key steps in the common soy processing process, which is the aqueous alcohol extraction process, leaching process. So we make modifications. Here show several key aspects in terms of the raw material and solvent selection and the drying stages. So we develop three strategies. The strategy one, we select low-cost starting material. So currently, the product use the water flakes, which are made with minimum or some level of heat treatment. This material is relatively expensive. So we select alternative low-cost, locally available material, such as default soy meal, toasted default soy meal, also the partially default soy cake from the EE process I just talked about. Then regardless of what the material, even though they are lower cost, the key is that this material should be in food grade because we are going to eat the product. Our second strategy is to choose alternative leaching solvents. Currently, they use alcohol, ethanol, as aqueous ethanol as a leaching solvent. So this can be in terms of the process, and you have a lot of environmental issue. They have the equipment got a much cost, have involved with desolventizing steps to remove the ethanol. So we choose the simplest and cheapest solvent is the water. This will process the alcohol-free process and can eliminate the desolventizing steps and environmental friendly process. Our strategy, third strategy, is to choose alternative drying method. Currently, the wet soy protein content is dried through the spray drying, which give a powdery products. So very the free fluid products. But the spray drying is good, but it's expensive. So we need to choose inexpensive and locally available drying methods. This method should have a lower initial capital cost, easy to operate, effective in drying. So example include like a rotated drum dryer, fluidized bed dryer, lower cost convective dryer, and solar dryers, or a combination of those drying methods. This chart shows the methods developed a few years ago for making lower cost soy protein concentrates. We start with soy meal, we just talk about raw material, extract soluble component with plain water, go through liquid solid separation. Then washing step is optional. Through washing, you use more water, but increase protein content in the final product. The drying is through the low cost drying method I just discussed. So because we will not use a spray drying, so the particle size of dry product may not be ideal for certain applications. So we can add additional step of grinding the product and make the powder. This is the visual appearance of two types of soy protein content. On the left is the commercially available soy protein concentrate, we call regular concentrate. This is through the alcohol leaching method and the spray dry product. On the right is a product made by the low cost method. You can see the color relatively a little bit brown. Here talk a few of this research done in the lab, in my laboratory. We use this method to apply this method to 4 types of soy meal or soy flake. The first on the left one is a defined soy flake. They have highest protein dispersibility index called PDI. The higher the PDI, the less the denaturation of protein. So 90% to 70 and 20%. Then we also use the lower fat soy flour made by the EE process and methods called extrusion expelling method. And the other flour have about 40% of PDI. So as you can see, the protein contents starting about 55% for the defined soy flour, partly defined about 52 because they still have a residual fat. But during this process, with a high PDI sample, actually the protein content is reduced because they're being extracted with the water, extracted out. But for rest, for 70, 20, and 40 PDI sample, the protein content are increased. In particular, for the last two items, toasted defined soy flour and low-refined soy flour, the protein content increased significantly. So the protein recovery in the final products also increased to about 75 to 80%. So we have to choose the right raw material with a lower PDI is better. And let's talk about how this process reduced the oligosaccharides. As you can see in the original soy meal, oligosaccharides, which expressed as raffinol equivalent. So our methods cannot distinguish between raffinol and stachyl. So they express as a raffinol equivalent. So all these four types of material, regardless of PDI value, they give similar reduction, almost around 95%. So it's very almost eliminated by this process. This table shows how the fatty acid get reduced by the process. Again, with this four type of soy meal as a raw material, the inner content around 2%, but through this process, we get about similar reduction around 60%, although not as high as oligosaccharides, but reduction in 60% is still significant. I think mostly because the fatty acid have some bonding with proteins. This chart shows the comparison for regular soy protein concentrates and the low-cost soy protein concentrate in several parameters. So protein content, the low-cost soy protein content may be slightly lower in protein content because they're around 65, but for regular one, minimum is 65%, some are about 70%. So nutritional value is almost similar in terms of oligosaccharide, fatty acid, or some other compositional attributes are similar. Then solubility and functionality is, as we can expect, they are much reduced because we use the meal that have been denatured. The product size we can get a similar because we can use additional grinding to get a similar fineness. The color is brown as shown in the picture. The flavor on the order, they may have some roast nutty flavor. In terms of applications, compared to regular soy protein concentrate, which have much higher, broader applications because they can be used for, because they're white color, they are more soluble functional, they can be used for beverage or some other application. But for this low cost soy protein application, they still relatively can be applied for quite a few products like porridge, breakfast cereals, snacks, bakery products, desserts. Also for meat patty, sausage, possibly for meat analogues. So they are for protein enrichment purpose or as a main ingredients for the product. So it can also be further processed into extruded soy protein product or textured soy protein product. So which is many used as for meat analog. Here they show some of these applications in different categories: the porridge, the bakery, breakfast cereal, meat patties, and snacks, and the sausages. Now this is the last slide. So hopefully for this presentation, you can get some takeaway message. So traditional soy foods appear less to many people. So soy protein products are widely used as an ingredient in various soy products, protein products, soy food product, variety of food products. So soy protein concentrate is better than soy flour, but it costs more with limited availability. So a few years ago at the US Department of Agriculture, we developed a method for making low-cost soy protein concentrate. The low-cost soy protein concentrate is brown in color and less water-soluble, but have same composition as a regular soy protein concentrate. It has a broad application in many local foods, and the method, I think, is adaptable in Sub-Saharan Africa region. This concludes my presentation.
Okay, thank you very much, Dr. Keshun Lu, for your presentation. And I just wanted to mention that questions to Dr. Liu will be posed during the panel discussion. In the interest of time, we'll just move right into the fourth and the last presentation for this session. So it's my pleasure to introduce the speaker for this presentation, Dr. Konstantina Kyriakopoulou. Dr. Kyriakopoulou is a postdoctoral fellow at the Wageningen University and Research in the Netherlands. Dr. Kyriakopoulou studied chemical engineering at the National Technical University of Athens in Greece, where she also completed her PhD studies. In 2017, she joined the food process engineering group at Wageningen, and there she is a postdoctoral researcher doing research in the area of sustainable fractionation of natural products for the recovery of functional and bioactive food ingredients. Her main focus is on plant-based proteins, particularly in understanding their functionality and exploring their structuring potential for the formulation of a next generation meat analogues. So Dr. Kyriakopoulou will be talking about challenges for next generation meat analogues. Welcome, and the floor is yours.
Thank you very much. Thank you very much for the nice introduction. So today I'm going to give you a short lecture about our research in Wageningen, and specifically in the Department of Food Process Engineering. We are working on developing high moisture extruded meat analogues, but mostly we are trying to understand how the biopolymers that are used in these applications behave and how we can actually adjust the ingredient production processes to achieve the maximum out of it. I'll start my presentation with my take-home message. So far we know that several plant-based ingredients are available on the market, and soy is one of the most common ingredients. These ingredients, however, are mostly, once they are produced and refined, are mostly aimed on general application. So you can see isolates and concentrates that can be applied in products like drinks, beverages, meat analogues, porridges, etc. But are they actually applied for? Can they actually be applied in meat analogues? Yes, they can, but usually we need to optimize the meat analog process in such a way based on the ingredients that are available. So this made us think, can we actually produce ingredients specifically targeted in this application? So optimizing the fractionation process to meet the meat analog application requirements. And to do so, we have to identify which functional properties actually of the proteins are important to produce this desired fibrous structure for these meat analogues. And our process should also consider to be sustainable and provide nutritional ingredients. What we know currently for the ingredient production processes, as also the previous speaker said, for many plant-based ingredients and soy specifically, which is a byproduct of the oil production process. Also, many other plant-based proteins are byproducts of another product. These are usually isolated, and the isolation process increases the value, nutritional value, but also the price of the product. In many cases, the industry is focused on increasing the purity of these ingredients. So at the end, you have pure ingredients with defined chemical composition that are stable products, and they also have a quite broad functionality. So for example, they are quite soluble, or they have desired functionality to be applied in multiple products. And what we actually see is that these pure ingredients are mixed together to produce new plant-based products like meat analogues and to achieve the desired structure. But through this refining process, a big amount of these ingredients is actually turning up to non-food applications, so for example, animal feed. So this made us think, do we actually need these really refined ingredients, and is this desirable for meat analogues? But before I go to answer this question, I want to give a short introduction about what a meat analog is. You've probably already heard this from the previous presenters, but meat analogues are products that they are usually based on plant ingredients, and they try to recreate the nutritional value, but also the aesthetics of meat. So try to mimic the meat texture. Usually there are two main, one commercial meat analog production process, which is the extrusion, but we also in Wageningen we produce the sear technology. The extrusion itself can be performed under low moisture conditions or high moisture conditions. When ingredients are treated under low moisture conditions, in fact, what is created is a texturized vegetable protein, which can be hydrated and kneaded into products like a beef burger or a beef patty. And with high moisture extrusion, what comes out of an extruder is usually products that look more like chicken chunks or small pieces of chicken meat. These products, as already said, are produced under high extrusion temperatures, but they give this and they try to recreate this structure of meat, which is highly desirable. Also in the same line, we have the sear cell technology, which was developed in Wageningen University in my department, the food process engineering. This technology also tries to mimic whole cut meat. And in fact, we are really interested in creating this fibrous structure and this juiciness of the product. The difference with the extrusion is on the size of the product. The product is quite bigger than actually what it comes out from an extruder dies. And our process is considered slightly milder in terms of temperature. And here in the pictures, you can see some of our products that we produced in our lab. And both of them are based on a mixture of soy protein isolate and gluten. And the top one, you can see it once colorants are added, how closer it resembles the meat muscle. So how does this technology actually work? We start with We start with the powder, the protein powder, which is the basis of our recipe. This is hydrated and feed it into our sear cell device. This type of device here is a quad device, so it's a cylinder which has a cylinder on the inside. Between the two cylinders, we feed the product, and the cylinders are heated up, and the inner cylinder is rotating. And with this rotation, it creates this desired fibrous structure. So it texturizes our protein. Once we open the device, we see the piece of slab, in fact, that comes out. So you can see here my colleague taking the slab out of the machine, and you can see how thick and long it can be. So cutting this slab into pieces, in fact, we can create, as my colleagues call it, a vegetable steak. This is, as I said, in a normal recipe, so soy protein concentrate and isolate and gluten. We are trying to understand how the ingredients actually behave, and here in this slide, I present you an overview of the recent research that has been done in our lab. So we test different ingredients that are available on the market. So we have the soy protein isolate and gluten, as I showed you before. These pieces are slightly smaller because they are made in the lab scale device that we have. What we see is that a combination of ingredients usually gives the nice fibrous structure and quite juicy product. You can have either two types of proteins mixed together. You can have either a protein or a carbohydrate mixed together, as it's seen here, which is soy protein isolate with pectin. We've also tested other types of proteins. So we see also pea protein isolate mixed with gluten that can also form fibers. Of course, they can be slightly different. In later research, we have also seen soy protein isolate mixed with flour containing oil also to form a fiber. But once we look at the really commercial samples, so if we compare soy protein concentrate and soy protein isolate, we see that the isolate itself, it's quite more rubbery and it doesn't form the desirable fiber structure as the protein concentrate. So this made us also look at the meat alternatives that are already on the market. And what we actually see is in general that the production of plant-based meat analogues uses a lot soy protein concentrates. So soy protein concentrates can give us quite fibrous structure, and while soy protein isolate on its own in any device, so in our sear device, but also in extrusion application, it doesn't seem to be a good ingredient. This product is, although soy protein isolate, more expensive and more concentrating proteins, so it can have some nutritional benefits compared to soy protein concentrate. However, this extensive purification procedure that it goes under, it doesn't make it a desired ingredient. So soy protein concentrate and water seem to be the best candidates for this type of products. And these are only produced by removing oil and some soluble ingredients. So this brings us to a conclusion that less purified ingredients are more suited for meat alternatives. If we look now how the fractionation process goes and compare the protein yield and the protein purity, starting from soybean with a relatively high for the soybean protein content, but trying to make it even more enriched in protein, just by milling or defatting, we get defatted soy flour, which already on its own has a protein purity around 40%. If we want to go further into the fractionation process by adding a washing step with, as the previous presenter said, with alcohol solution, we see that we create protein concentrates where the protein purity is increased around 65%, but immediately the protein yield drops. And the more intensive the fractionation steps go, the more we can increase the protein purity, reaching up to 90%. But all in all, at this last process, we lose about 50% of the actual protein. And this usually goes to non-food applications, so we cannot recover this. So this actually made us think, how can we create functional fractions instead of isolates or isolated components, for example, and high protein isolate and some carbohydrates that we mix together to create the meat analogues, instead of really fractionating and mixing in high pure components together to create functional fractions. In this way, we can take advantage the richness of the matrix and avoid over-processing. And in the process, we had as a goal to understand what are the key behaviors of ingredients that we need for meat analogues, and based on this, suggest a reverse engineering approach on how to sort of reconsider the fractionation process to create ingredients for our application. To start, we first saw the traditional fractionation process to create a protein isolate. And what is the reasoning behind the selected steps that are there? So we start with the soybeans. We mill the soybeans, defat them to remove the oil, which is actually the main product, as I said, of the soybeans. Afterwards, an alkaline solubilization step is introduced. This increases the solubility of the protein, disentangles the protein from the other components, and in that way, we can remove any insoluble carbohydrates. After the solubilization step, we have an acid precipitation step where the pH is adjusted close to the pI of the soy protein. So we can actually separate a significant fraction of the proteins. After in this step also, we remove any soluble carbohydrates. And after this step, we can wash our product and neutralize it in a neutral pH. And this neutralization step actually helps significantly the functionality of the protein. And at pH 7, the solubility of the protein in general is relatively high. So that's how a protein isolate is recovered. In our process now, we suggest an aqueous fractionation process where actually the defatting step which is usually with organic solvent doesn't take place. And we remove the excessive washing steps because, in fact, we don't want to purify our ingredients so much that they don't work for our application, as I showed you before, so for the soy protein isolate. So we created, in fact, a mild fractionated soy protein, which eventually had a protein content of 75% with an index of a conversion factor of 5.7. We see also some oil still remaining in the sample, although we didn't use any organic solvents. However, a significant amount of oil was removed, so the defatting process can also be considered successful. As I said, the enrichment is quite high, but we still have some carbohydrates remaining in our fraction, which are beneficial. We tested this product in our Circel equipment, and we compared this product with a protein concentrate and a soy protein isolate. We mixed our product with full-fat flour to actually recreate the composition of the soy protein concentrate, which we knew that was a successful ingredient. But in fact, after the structuring in our Circel device, we didn't see any fibers. Although the sample was quite juicy, no fibers were seen. And what we decided to do is to actually toast our ingredients. So we applied a heating step after the fractionation, and we used this material and we saw that fibers started to appear. So in fact, even if you have exactly the same composition, this doesn't guarantee that the product will work. But we see with small treatment steps, like post-treatments by heating, for example, you can create such functionality that can yield some desired fibers. Already by mixing the ingredients with water, we could see some differences. So you can see the soy protein isolate, how it behaves, the concentrate. The non-toasted, it was more like a gooey dough instead of a crumbly dough, but the toasted one resembled very well the soy protein concentrate. The way that the product sort of holds water, or if it can be solubilized, affects these properties, and that's why we tested the water holding capacity and the nitrogen solubility index, so to test how much protein is actually solubilized. And we see that the successful samples for creating these fiber structures, so sample number 2 and sample number 4, had sort of intermediate water holding capacity, intermediate to high, and intermediate nitrogen solubility index, or intermediate protein solubility, while the non-toasted couldn't hold so much water. So maybe that's also what we see in the structure here. From this research, we concluded that distinct fiber structures could obtain when the soy protein isolate was thermally heated at 150 degrees. And together with the combination of native flour, showed that not so refined fractions could yield this desirable fibrousness. This means that maybe we should focus on not completely purifying our ingredients, because also our functionalities are not the classical functionality, so we are not looking also for high soluble products, for example. By avoiding intensified extraction steps, we can also make our product more sustainable and keep inside the material ingredients that could be beneficial. And post-treatment steps can be a way to actually modify the ingredients to the desirable, for example, water holding capacity and solubility. So by heating, we can increase the water holding capacity and reduce the solubility in that extent that it creates a really functional ingredient. And this research is still going on, and we still working on understanding how the properties of the ingredients behave. And we designed different fractionation process and dedicated forming analogues. So from the aqueous fractionation process that it's already introduced, we also thought about changing some steps, so going to even a simpler fractionation process by removing, for example, the acid precipitation step, or even removing the alkaline solubilization step. And what we actually observed is that the composition was significantly different. So we have still enrichment on the protein, but by removing the acid precipitation step, the enrichment is lower, so it's closer to a protein concentrate. But in this fraction, still a significant portion of soluble carbohydrates remain together with the protein, while in the last line, the yellow line, while we have a bit lower protein enrichment, we have significantly higher oil enrichment, and insoluble carbohydrates still remain in the fraction. So by selecting the right extraction steps, we can modify significantly the composition of our ingredients. We also went one step further, so not only modifying the composition, but modifying the functionality and the nutritional quality. And in fact, this we did by just replacing the sodium hydroxide in the fractionation process by calcium hydroxide. In this way, our goal was to actually produce high calcium, low sodium soy protein ingredients. And by doing this change between calcium, sodium hydroxide and calcium hydroxide, we actually also saw a significant difference in the functionality. And why are we interested really in replacing sodium with calcium? So calcium on its own, it's a nutritional component for the human body. It helps on the strengthening the bones, the human bones, but also we see that with the changing from the animal-centric diet to a plant-forward diet, maybe a significant amount of population will experience some calcium deficiency. So enriching the products with calcium can be beneficial. On the other hand, so for sodium, we see that especially the Western diet, overconsumption of sodium is observed. And we know that sodium is added also as a flavorant, but also it has some technical functionality, and that's the reason that you see it in many products. But we also understand that sodium is introduced in the ingredients, the plant-based ingredients, through the fractionation process. So in fact, through this alkaline solubilization step and neutralization step, you add actually extra sodium in your fraction. Here I have a comparison between a piece of meat. So the content of sodium in a piece of meat, for 100 grams of meat unmarinated without any additional taste components, you have approximately 100 milligrams of sodium. While a corresponding piece of meat, like a vegetarian steak, just using soy protein isolate, we are talking about a high moisture content product where you only need 30 grams of soy protein isolate to create this structure. You get about 3 times more sodium. So you already see that by using these ingredients, you add extra sodium in your diet. So we looked at the same fractionation procedure by only changing the sodium hydroxide in the solubilization step or in the neutralization step with calcium hydroxide, or in both steps with calcium hydroxide. What we actually observed is that the sodium content, when the calcium hydroxide was used in the neutralization step, the sodium content was significantly lower, which is something that we are interested in, and the calcium content was enriched. But at the same time, this enrichment in calcium caused some changes in the functionality of this soy protein isolate. And this can be seen in this graph. So in fact, I plotted here the water holding capacity, so how much water the ingredients can take up, and also the solubility. So the original fraction closer to the soy protein isolate, where sodium is used in the alkaline step and the neutralization step, has a really high solubility. While we increase the calcium content, we see that the solubility of the ingredients start to increase significantly, and especially when the calcium hydroxide was used in the neutralization step. So we can, by changing the calcium content in the product, we can already modify the solubility, protein solubility of our ingredient. In these ingredients now that have a really high solubility index, if we apply a toasting step or another heating step, we see that this actually increases the water holding capacity, and also decreases the solubility. So the same can happen in any ingredients with a heating step. And if we look at the desired properties for the meat analogues, as I said from our the first part of my presentation, we see that medium solubility it's more desired for this application, but also medium to high water holding capacity, it's also a plus, but not too high as we had for the soy protein isolate, the commercial sample. So we, by creating these functionality maps, we try to get insights about which properties are more relevant for our application. And this brings me to my final slide, which is again take-home message. We are researching the key functionalities of the proteins, and we dedicated for fiber structure formation for meat analogues, processed in extrusion process or the sear cell technology. And we see that the functionalities that are desired are quite different of what the commercial products are available have. So, for example, we are not looking for really high soluble fractions. I've shown in my presentation that by changing the fractionation process, we can improve the nutritional quality. So we can enrich, for example, the fractions with calcium, or we can even reduce the sodium content. And by changing the processing, we can improve the functionality, or I would say tailor the functionality towards our desired range. And at the same time, by removing any excess steps, we can also play a role on sustainability. For example, removing any excessive washing steps that are dedicated for purifying the ingredients, which we actually don't need. By putting together all this information, we can we can focus on creating a reverse engineering approach where we can design our fractionation process so that the ingredients produced can fit to our application. And with this, I conclude my presentation. This is my contact details, and I'm available for any questions, and you can contact me through my email.
Okay, thank you very much, Dr. Kyriakopoulou, for your excellent presentation. I really enjoyed it. Without further ado, I think we'll move straight into the panel discussion. Then the audience will get a chance to ask questions to Dr. Kyriakopoulou, but also to the other speakers at the presentation, at the session. So I'll briefly recap the members of the panel, the presenters for this session, starting from Dr. Stacie Dobson and Dr. Alejandro Marangoni, who is not able to join us now, but Stacie is here. And we also have Michelle Braun, Dr. Braun, and Dr. Keshun and Dr. Konstantina Kyriakopoulou. All right, so I will start with questions for Dr. Liu, and then Dr. Kyriakopoulou, okay? So you have a question from the audience, Dr. Liu. When you say the solubility is reduced for the soy protein product, does this mean that the taste is grainy?
solubility. So we use the starting material will be denatured. So the protein will be denatured in the product. So solubility of protein is very low. That there's nothing to do with, you talk about green, right? Bini, you talk about green or you talk about flavor? What the question? Can you?
The question is, when you say the solubility is reduced, does it mean that the taste is grainy?
Oh, grainy. Oh, yes. Okay, I see. Oh, grainy. Well, this is depend on not for beverage. So this is not for beverage. So that means the grainy and the solubility are not totally related, because grainy relate to particle size of products. So when we, after this drying, we reduce the particle size, and then incorporate into select or bakery. They are similar particle size to as other ingredients. So they are, I don't think they will cause a grainy little bit. It's a good question. But the protein solubility, see, when doing this bakery or the product, their protein solubility reduced anyway. So I don't think they affect the grainy texture. So it depends on particle size.
Okay, thank you very much, Dr. Liu. So the next question is for Dr. Kyriakopoulou. What is the fate of oil in the alkaline solubilization of full-fat soy?
Yeah, that's a really good question. So by using immediately from the full-fat flour, the alkaline solution, in fact, the oil comes out in a creamy layer. So, and in that layer, it's probably in the form of oil bodies. So we see a potential for this material to be used as an emulsifier immediately, as condensed and be used as a sort of vegan mayonnaise. But also, if we want to really separate the oil, it's possible to break these oil bodies and separate the oil by intensive centrifugation. Probably you can also take out the oil. But for our product, as already said in the previous presentation, the addition of oil in the meat analogues is really difficult. The oil tends to separate. So by using ingredients that already in the natural structure, they contain part of the oil, we consider that really important.
Okay, thank you very much, Konstantina. So I have a question for the panel, but this for the general panel. So maybe if you have the expertise, you could answer. What is the future, in terms of plant protein processing? Because from the presentations, you see that sometimes when you apply processing, you compromise some functionality. So what do you see as the future in terms of minimal processing to plant-based proteins that can be used to achieve maximum functional and health benefits in food products? So, any one of you can answer, if you will.
I can take that first. That's no problem, if you don't mind. Thank you for the question. And your phrasing was, talking about processing. And I think a point that I conveyed within my talk was processing doesn't always take away nutrients, right? Sometimes it concentrates or isolates them more, makes them more digestible, more readily available. So we have to be mindful in the processing decisions that we make to optimize the nutrients. And we heard a lot of different strategies throughout the course of this session for doing so. There are many other strategies that are currently in place that we didn't get to touch on. And this is going to be an exciting area going forward of how can we take the plants that the producers, the farmers have become so good at growing and harvesting, and take that forward to help to feed our growing global population and supply that essential nutrient protein. And since I come from the nutrition perspective, you're talking about the future state, I think it's exciting as we continue to learn more about this source, soy, that's been in the food supply for so long. We're still learning of the health effects that it may have in terms of some of the conditions that we face currently, in terms of threats to our health. The challenges, the nutrition challenges, the health challenges that we face today are different than those that we faced, say, 50 years ago. We were doing research on soy back then, but for different outcomes. So there's still a lot of work to be done. So I will pause there and let someone else.
Okay, thank you, Michelle. I'm not sure if anybody else wants to add to that, because we're now receiving some more questions. So maybe I can pose more questions, and maybe you can jump in if you have comments on this question. You can add it then. Okay. So this next question is also for all of you. So besides soy, are there other plant proteins that can be economically used to mimic animal-based proteins? So maybe Stacie can start.
Yeah, I can jump in on that. I think that soy has been existing as that main protein for a while. Personally speaking, for the project that I've been working on, we've actually chosen to use pea as our main source, as our main protein source, because it is offering that alternative protein other than soy, as depending on the environment, people do try to steer away from it sometimes, but it does have great structural properties, soy protein does. But I think in the future, we'll see other bean proteins or other legume proteins being looked into further, like chickpea, lentil, and understanding their functionality more as we try to determine what all these other types of kind of plant proteins can do for us.
Maybe since you just addressed this question, there was another question specifically for you, Stacie. So maybe you can continue. It says, can prolamine chelate minerals like iron?
To be honest, I haven't looked into it. There may be some possibilities. It's an incredibly hydrophobic protein in the sense that it doesn't, when you mix it with water just plain, it doesn't dissolve. You have to work with it to get it to make its fibrous component. So I haven't explored using any type of chelating metals, but we found that our organic acid worked best for us to get that extendability. But using it in another function, you may be able to use those type of salts.
Okay, so is there any other take on this? So I was thinking that this question is also maybe related to the use of the protein metal chelate as a means of delivering the mineral for mitigating micronutrient deficiency. So looking at it from that perspective, maybe Michelle or Keshun, so have you seen any examples of using proteins, particularly prolamins, as a mineral chelator for delivery purposes?
Mineral chelate is you talk about for texture or for mineral chelate for this kind of protein?
Well, there is no particular applications here, but from Dr. Braun's presentation, proteins also were presented as also a means of enhancing mineral absorption and reducing micronutrient deficiency, like iron or zinc deficiency. So looking at it from that perspective, especially in food product formulations in at-risk populations for micronutrient deficiency, do you see any possibility of using these proteins, prolamins, to chelate mineral?
I think for Dr. Braun probably better. He's works for nutrition area. Maybe Dr. Braun can answer this question.
Okay. Dr. Braun?
Right. Iron absorption from plant sources can be a challenge. It's not as optimized from the plant sources as it may be in other forms. So when I mentioned the product that was fortified with iron, we have to think about the form of iron that's available as well. We're approaching this from the protein side of things in this question, but we also have to think about the form of the iron. And then in considering overcoming absorption challenges, other anti-nutritionals that may impact absorption, a lot of times those minerals are added at a higher level to the plant sources to help to overcome those absorption drawbacks. So in the product that I demonstrated, there was a higher amount of iron available in that product, just to ensure that the iron is at some proportion absorbed by the consumer.
Okay, thank you very much, Dr. Braun. So I have another question here for everybody. Have other phytochemicals been added to soy protein / analog products? So we already talked about iron now. So are there cases where other phytochemicals are added to some of these texturized protein products?
You talk about other ingredients or
other phytochemicals, yes.
Oh.
Other compounds for fortification purposes, like vitamins or other minerals for food fortification. So are there other cases where other phytochemicals have been added to these products? So in addition to just creating the product functionality, can you also add other phytochemicals?
Yeah, I think for middle analog during this extrusion, by extrusion, like talk about high moisture extrusion or even the low moisture, during this final products, they probably can add some vitamin. But during this before process, they cannot be added because the temperature, like for vitamin destruction, may be high. So, but after this, it's a post-extrusion process. Probably they can add some of this phytochemicals, I think. I think for maybe other speaker, maybe you could, because they all deal with kind of products. They probably can add some more.
Right, thank you, Dr. Liu. I think Dr. Kyriakopoulou, maybe you could weigh in here, since you talked about using calcium hydroxide for isolation of the proteins.
Yes,
so maybe it,
we can see for these meta-analogues that are on the market that there is also a trend, especially for the ones that are basically for vegan consumers. There is a trend to fortify them with vitamins and iron, just to meet the requirements for people that don't eat, for example, red meat. So that's also a possibility. And in many cases, I would say the combination of proteins that are used, not only in terms of structuring, but also adding additional proteins in the product as binders or maybe flavoring agents or have other functionality. They can also add amino acids that are missing. So to make the amino acid balanced, the product balanced in amino acids.
Okay, thank you. So speaking about adding amino acids, sorry, go ahead, Michelle.
I was just going to add, we heard a lot about calcium, different salts that can be used. Potassium is another commonly used salt in the processing. And so, there's in the process, it can be added, but I believe the question was, what other fortifying nutrients can be present? And a great example we can look to is infant formula. So, that's a foundational product that's available, that's the only plant source that has been demonstrated to be viable within that. The delivery system is soy-based infant formula, where it meets the nutrient needs, macronutrient needs, as well as micronutrient needs, and a full complement of vitamins and minerals and other key nutrients to help to make that product as close to breast milk as possible, and definitely analogous to cow's milk formula, which has been demonstrated in studies. So, it begins from that infant formula format, but certainly a variety of vitamins and minerals can be added to these products. And the ready-to-use therapeutic food that I mentioned also had a matching vitamin mineral premix, similar to the peanut and cow's milk based product, which also have vitamins and minerals added. Sorry, over to your next question. Just wanted to add that point.
All right, thank you, Dr. Braun. So this next question is specifically for Dr. Kyriakopoulou about the full fat soy protein isolation. So the question is about the oxidative stability of this product. So what do you think, for the products that have high oil content, the protein isolates that have, or concentrates that have high oil oil content? Wouldn't there be issues of oil oxidation, lipid oxidation during prolonged storage?
So we've seen that the state in which the oil is stored, so if it's in the original oil bodies, it's still protected. So this doesn't show any oil oxidation. But once you have intensive processing and you break these oil bodies and the oil remains in the sample, we didn't test it exactly, but we have evidence with some smell, let's say, coming out of samples that have been stored for a longer time that show this oxidation. But yeah, this is ongoing research, and we want to see how stable these products are longer as food ingredients that you store for a longer period of time.
Okay, thank you for that answer. So I have another question, maybe two more questions, but one is for everybody. I just want to get a perspective in terms of cultural sustainability. Since we're trying to develop protein-based products that is acceptable across different populations in the world. Now, what do you think about the type of processing that you need to do to achieve a product that has taste and texture and other properties that the consumer would accept, because you're trying to use it, especially for texturized proteins for meat analog applications. So how do you achieve a product that is culturally acceptable to the consumer in terms of taste and texture?
I can take a first stab at that. Go ahead.
Oh, okay. Yeah, I think this is they're durable, but depend on the cost. I mean, everything is can talk about what kind of process, what kind of equipment they use. So also the local taste and flavor. All this is kind of during these product development stages. So they are durable, but depend on what the cost of what product and the development, right? What kind of ingredients. Let's say you want to make a meat out of what kind of the textures, protein product you want to use as a starting material, or even during initial selection stages. Like during the high moisture extrusion, you can start with what kind of raw material. So there are many choices in terms of the ingredient selection, the processing equipment, and after process, you also what kind of flavoring, the formulations, all this kind of all work together. So there's no single answer on this one. It's durable, but depending on what the cost you want, you can absorb.
Okay, thank you, Keshun. Michelle, you were going to add your perspective.
Sure. Just to build on that, it's important to think about the quantity or the dose, the amount of protein that you want to deliver in that final product. And it comes down to too much of a good thing can deliver off notes or off flavor. So when we go to higher doses of any protein, then those characteristic off notes come through. For soy, it's beany. Pea has its own off notes. Dairy has its own off notes. It's barnyard is how it's characterized. And so sometimes blending proteins can help to optimize or still reach those target doses with minimizing the off notes that come from just one protein source. But the question was about how these protein sources can be a part of traditional diets. And I think it's first important to acknowledge that tastes are very regional. So that beany flavor is very much preferred in regions such as Asia Pacific. Those are not as necessarily is seen as off notes as much as they are in other parts of the world, whether it's desired for a savory flavor or a sweet flavor that gets added to a finished product, again, it's very regional specific. And when we do sensory work, we don't extrapolate what we learn in one country or one region to the rest of the globe. Instead, we do those studies appropriately in the regions that have the users, the tasters, that will receive the product in the end. So it's important to take those elements into consideration.
Okay, thank you, Michelle. So Konstantina and Stacie, do you have perspectives to add?
I can touch on it just briefly. I think you had mentioned about the cost involved in these productions, and as we've seen, some with there's the shear cell, it can create great fibers, and extrusion can create great fibers, but sometimes those means aren't as feasible in parts of the world as they are in others. So I think there needs to be more exploration and methods that can be done kind of without these type of equipment. That's what I've been working on, as I think making this product that doesn't involve all this extensive equipment kind of makes it more adaptable in places that can't afford it, and also trying to explore the elements of adding different types of protein in it, so you can kind of customize it based on which region is growing certain types of plant-based proteins, and trying to develop the product for what they have. So kind of creating a system that can be applied. I think as the plant-based industry continues to grow, it's definitely something that I can see it building into.
Okay, thank you, Stacie. And Konstantina, if you have a perspective, I would like you to combine it with these specific questions targeted to you. It says, what is the advantage of your process over high moisture extrusion for your protein isolation?
Yeah. So for the previous question, I would also say to really consider not only the protein enrichment that we want to achieve, like to not overprocess the ingredients. And we can also see that going to more traditional methods like fermentation, you can achieve some enrichment and in parallel some removal of anti-nutritional factors, which can be beneficial, and it can also improve the flavor for the countries that cannot accept this beany flavor for the product. So I think there are many ways to really modify the ingredients without over-processing, and these ingredients can be incorporated in the diets of the different people. And for the second question, so the CIRCEL technology is developed, it's sort of in parallel with the extrusion process. The difference is it's a batch process. And in fact, we don't have so much mechanical energy given into the system, but we still can achieve really fiber structure. And the system that we have, this quet system, the cylinder in a cylinder, can allow us the development of really bigger pieces of product. So instead of being limited, like in extrusion, in the die morphology, how the product comes out of the extruder, our product, it sort of takes the shape of the cylinder, and it's much bigger in size. And so far, the ingredients that we use seem to behave, let's say, form fibers in temperatures slightly lower than the extrusion process. And we are working even further to reducing this the need for high temperature.
Okay, thank you very much, Konstantina. And this question brings us to the end of the panel discussion. And before we close, I would like to take another moment to thank you, the speakers, Dr. Liu, Dr. Braun, Dr. Kyriakopoulou, and Stacie Dobson. Thank you very much for your excellent presentations on innovations in plant protein processing. So this has been a very wonderful connection between fundamental scientific research and consumer-targeted applications in enhancing functionality and quality. So from these presentations, I see a very bright future in translating proteins from plant-based food, especially soybean, but other legumes and cereals as well, into value-added applications. And it's a pleasure that you have presented your perspectives and your innovative research in this direction. I would also like to thank the audience from wherever you have joined us. Thank you very much for participating in a Soybean 360 Symposium. And I hope you were also able to join the rest of the 3 days of this symposium. Thank you very much, and bye-bye.